Okay, so maybe I invite Mr. Bilal Ahmed But to give the opening remarks first, please.
Yeah. So if all our participants are there, then I may start with the permission of all of you all are my seniors in that context, very accomplished scientists on from both sides. So let me take the opportunity to start this event. Bismillahir-Rahmanir-Rahim. Assalamu alaikum. Good afternoon, ladies and gentlemen. At the onset, I would extend my gratitude to Dr. Lam, Director of State Key Laboratory of Agrobiotechnology, Chinese University of Hong Kong, for arranging this webinar on very important subject of agriculture environment interaction and its implications. I'm also thankful to Dr. Muhammad Anjum, Director General Agriculture Extension Punjab, and our worthy speakers from MNS University of Agriculture, Multan, Ayub Agriculture Research Institute, Faisalabad, PMAS Arid Agriculture University, Rawalpindi, and University of Agriculture, Faisalabad. Dr. Lam, these are very renowned universities working in the field of agriculture, and we are really very honored and privileged that speakers from all these universities are here. These are very huge universities working exclusively in the field of agriculture. Let me admit that I have always learned from Dr. Anjum and other worthy academicians about the issues confronted in by the agriculture sector in Pakistan, and during the public policy, whenever I needed to learn and to take measures that could be taken to address those challenges, I learned from our worthy colleagues all the time. Pakistan is a country with a population of 220 million people with an area of about 800,000 square kilometers. However, less than 30% of land is being cultivated. Though Pakistan is 4th largest producer of cotton, 8th largest producer of wheat, 5th largest producer of sugarcane, 13th largest producer of rice, and 25th largest producer of maize. But unfortunately, productivity of Pakistan is much less than desired. Productivity of all of our major crops is in the middle of the continuum of major producer in the world. Whereas I have found that mainland China is placed normally at the top or at the second position in terms of productivity globally. Of course, there are several reasons that can be attributed towards the low productivity, like poor quality seed, lack of modern irrigation systems, lack of knowledge about balanced use of fertilizers and pesticides, and absence of culture of corporate farming in Pakistan. But in order to feed such a large population with a small land area, there is a dire need to enhance the productivity of crops in Pakistan. Fortunately, we have, as I mentioned, that fortunately we have large body of scholars having expertise in various fields of agriculture. This is a plus point which can be leveraged. However, through collaboration between Pakistani scientists and our friends from mainland China and Hong Kong, this the body of knowledge which our scientists possess, that can be leveraged tremendously. We can learn from the experiences and expertise, the way the Chinese friends have achieved the level of productivity. We really would like to learn from Dr. Lam, your team, and your institution. For my friends from China, Chinese University of Hong Kong ranks among the top 50 university of the world. It was 39th in QS ranking recently. Asim, it was 39th in, yeah. And it has produced several Nobel laureates. You might be surprised to learn that there are several Nobel laureates from this prestigious university in the various fields of learning. The excellence of research and development of Chinese University of Hong Kong has been acknowledged globally and termed as its hallmark. So this webinar basically has been arranged to initiate a process of dialogue for mutual understanding and learning, and to explore further areas of mutual cooperation. I am confident that tremendous potential for cooperation exists between the two sides. I am also confident that this interaction will open doors for further cooperation and collaboration in various fields of agriculture. I would congratulate Professor Lam, Director, State Key Laboratory of Agrobiotechnology, Dr. Muhammad Anjam, Director General Agriculture Research, and Mr. Asim Malik, Consulate General of Pakistan, for organizing this much needed event. However, it may just be the beginning of the cooperation between CUHK and Pakistani counterparts. I would really be looking forward to participate and arrange such kind of joint event in future to deepen our friendship, cooperation, and to enhance mutual learning. So thank you very much. Over to you, sir, Dr. Lam.
Thank you. So next, may I invite Dr. Mohammed Anjum to say a few words.
It's indeed a matter of great honor for me to welcome our friendly country, Hong Kong scientists, specifically starting from Mr. Bilal Ahmed Bhatt, Consul General of Pakistan, Professor Hong Ming Lam, the Director, Key Laboratory of Agrobiotechnology. This is really a matter of honor for me to participate in this joint discussion on agriculture, climate, and its implications. Basically, if we're talking about the climate change and the agriculture, Punjab being a major province in Pakistan, having over 70% share in agriculture, and having the surpluses in wheat, rice, sugarcane, gram, even fruits and vegetables, is highly vulnerable to the climate change. We are facing the hot and cool temperature threats. We are facing the high and low rainfall threats. We are having the problem of the pest outbreaks, the transformation of secondary to the primary pests, the invasive pests, the seed abortion problems, the germination problems, and the yield reduction problems in rainfed agriculture. We are having almost the climate shift situation in Pakistan, not the climate change. When we are saying the climate shift, this monsoon or the traditional rainfall pattern in Pakistan is completely disturbed. We are having the perennial surface irrigation supplies, and that is from the Indus River and coming from the glaciers. But the dominance in the surface supplies is from the rainfall patterns, from the 4 major rivers flowing from Punjab province. These 4 rivers are giving the intermittent supplies. That's why in the critical stages, we are facing a great water shortage issues. This time in the Rabi season, we are having 2 major crops. One is wheat and the second is gram. Gram is grown on over 2 million acres. We are harvesting 0.7 million ton to 0.2 million ton on a year-to-year basis. If we are getting the good rainfall in the month of October for the rainfed area, because the gram is grown in the low rainfall areas, we are having a good crop. But if we are getting the low rainfall, we are having the low productivity. All this is basically can be addressed through the biotechnology. And biotechnology is now a game changer. We are fortunate that we are having a great scientist from our friendly country, Hong Kong, and Mr. Bilal Ahmed Bhatt had made this possible to learn from our colleagues from the Hong Kong University. And likewise, we are having our own scientists from Pakistan who are having an experience to talk about the climate change. But we will be having this roundtable discussion to go for the concrete recommendations for best of Punjab agriculture, which is, I have earlier said that over 70% of Pakistan agriculture. We are having distant ecological zones. We have 10 distant ecological zones where we are having the rainfall from 50 millimeter to 2000 millimeter, and each ecological zone has a different temperature regimes, different cropping systems, and everything is possible with the technology. We are very much eager to adopt, acquire, to get and acclimatize these technologies in Pakistan for our better seeds, for our better agronomy, for harvesting, for post-harvesting, for mechanization, and this is possible through the new sciences. And at the outset, I again welcome the Consul General of Pakistan for this endeavor. Again, pay thanks and profound gratitude from myself to my Hong Kong University colleagues and likewise Pakistani colleagues to participate in this beautiful discussion. At the end of the discussion, I'm pretty much sure that we will be learning a lot, we will be exchanging the ideas, and that ideas will come to the win-win situation from both sides. Thank you, sir. Thank you, Bilal. Thank you, Professor. Thank you, sir.
Thank you. So maybe I say a few words on behalf of the university before I move on to the presentation session. So dear Consul General, Mr. Bilal Ahmed and Dr. Mohammed Anjum, distinguished speakers from Pakistan, and dear colleagues from our university. So on behalf of the State Key Laboratories of Agrobiotechnology at the Chinese University of Hong Kong and the RDC AoE Center for Genomic Studies on Plant-Environment Interaction for Sustainable Agriculture and Food Security, I would like to welcome you all to the CUHK-Pakistan webinar on agricultural environment interaction and its implications. The webinar would not be successful without the dedicated preaching effort from the Consulate General of Pakistan in Hong Kong and the Office of Academic Link of our university. Thank you very much. I hope that research collaboration between both sides could be nurtured through this high-level scientific exchange. The Hong Kong team has been pursuing research excellence and achieved some groundbreaking research outputs in genome sequencing, reference genome building, as well as novel technology and protocol establishment, etc. We have introduced a green economy scientific industrial partnership model for stress-tolerant soybean cultivations, which has been shown successful in Northwest China. These efforts contribute to the United Nations Sustainable Goals, including low poverty, zero hunger, and climate action. On the other hand, we have already established an extensive international collaboration network to practice to extend our platforms for sustainable agriculture from Hong Kong to China and to the world. Pakistan certainly will be one of our major partners. The world is facing a climate emergency. Rapid urbanization and the growth of megacities has posed complex challenges for environmental sustainability development. To combat food security and climate change issues, agriculture-environment interaction would be a major area of attention to tackle agricultural tasks, including yield enhancement, quality improvement, stress tolerance, and low-input sustainable agriculture. Leveraging our team's strength in the area of research in environment and sustainability, we are enthusiastic to match complementary expertise in Pakistan and foster strategic collaborations to address the challenges on climate resilience and sustainable agriculture from a global perspective. Last but not the least, thank you all for taking time today. I wish we all have a successful and fruitful academic exchange in the webinar. So before we start the presentation session, may I invite everyone to take a picture. So turn on your camera, and my assistant, Kathy, will help to take a photo for all of us. Thank you, Kathy.
Yeah.
Okay, so I'm the first one to give the presentation. So our team is the State Laboratories of Agrobiotechnology at the Chinese University of Hong Kong. And this laboratory is a name given by the Ministry of Science and Technology in China. So our team has 24 member labs from 3 universities with different age groups, so that we have our own sustainability. So here are the members. So I invited some of the members to join today. They represent different kinds of technology that we are using in this team. There are 4 strategic research areas, and one of them is related to soybean research that is led by my team. So how to approach the soybean research and eventually making use of it. So we start with the genomic technology and making use of the seed resources in China and the world. Seed is very important, but how to make best use of the seed is to use the genomic technology to understand the types of seeds, the mechanism behind the behavior, and use markers to assist speeding. So we have joined a international effort to advocate this kind of new breeding strategies, which has been published recently. So in brief, we have seed resources all over the world. Making use of the genome information, it will speed up the speeding and enable us to put the traits together and maybe stack it up so that we can apply it to the field. We have done an extensive check of the world's lagoons seed centers and found that actually there's a lot of seeds available in different parts of the world. And those are very important resources that we can use genome studies to analyze. Soybean is my interest, and together the world have more than 90,000 types of soybean that we can use for breeding. So we use the whole genome sequencing to do several things. First is to study the high biodiversity of wild soybean, because the domesticated soybean has a narrow genetic background due to the bottleneck, while those in the wild environment retain most of the genes and alleles that could be used to adapt to climate change. So making use of the genome information, we have more than 10 years ago, we are the pioneer to use this technology to investigate Chinese wild soybean and show that they have a high genome diversity. Subsequently, we join force and collect several different soybean studies. So this is an example showing my collaboration with Korea. So Korea also have a lot of collection of wild soybeans. So we use the genomic platform and helping them to study their collection, which mainly come from Korea, Japan, China, and Russia. I also joined force with China, mainland China, to investigate the most popularized soybean in China in the last 100 years. There's a total of 134 that has very large area of applications. However, we found that 14 of them will be able to capture 95% of the variations. What does it mean? It means that the genetic background is very narrow for these big soybean cultivars. So that also means that making use of the seed resources, there's a lot of room for further improvement of soybean in China. We also joined force with the USDA to help to finish the 1,000 soybean genome projects there. And of course, they have a lot of collection originally coming from Asia. Besides to study the variation, we also want to make efforts to have the world work together to improve soybean. So we published the first reference grade genome of wild soybean, and this is an international effort, and we release this data to everybody to use. So people studying wild soybean now will have a good reference. Now, besides those academic studies, we want to see how we could go from laboratories to the field. So this is the field I'm talking about. China has some good lands in northeast China, but in the northwest China, it's very dry. As you can see, the arid examic area here in my picture. And in some regions, they have salinity problems. So the white color are salt. This is me. So this is the land that very difficult to have cultivation. So China only have about 26% of woodlands. All the others are deteriorated. So it's very important to do environmental friendly agriculture. So one research I did is instead of just sequencing to understand the variations and diversity, we tried to using the genome technology to identify useful gene. One of them is a salt tolerance gene. So this is a salt tolerance wild soybean, which is salt tolerance, salt sensitive cultivated soybean. You can see the big difference here. So we successfully find the causal gene from the wild soybean. And subsequently, we use this gene as a marker to screen for cultivated soybean that containing these salt tolerance genes. And we got 3 cultivars successfully screened and tested in northwest China and receive certificates from the local government so that we can give it to the farmer to use. And we're aiming at using these new cultivars to increase income of the farmer and soil quality of their land. And here our soybean growing in very dry area. Here are the farmer who have harvested soybean from that was originated from our soybean cultivar. So we also practice different way of growing soybean. One very successful method is to do intercropping without sacrificing whatever are growing on the field. We just ask the farmer to grow additional soybean to replenish the soil with nitrogen and to increase their income. So our soybean can go well with wheat, flax, corn, and trees, proper trees, and apple trees. So this could be a very important application of our soybean. And we also try to see if our soybean can go in very drastic environment and use it as a pioneer crop to restore abandoned fields. And the results are quite encouraging. Recently, we also developed semi-wild soybean for forage so that the animal can eat the high biomass resulting from cultivation of soybean. So in order to connect to the international community in these pandemic seasons, we have started to organize monthly agro-biotechnology talk. Each month, I invited experts to talk to everybody in the world, if anyone can join. So far, we have 15 already. So we go for 15 talk, and there are some more coming. So if the friends from Pakistan are interested, you're very welcome to join our monthly talk series, which will cover different topics. Here is the YouTube channel that you can visit the back issues. So each talk we have videotape it so that just in case you miss it, you can listen and watch back the video to understand the topic. So that is what I will want to share for today. And so let me invite the next speaker then, right? So the next speaker, let me do a little introduction. So Professor Dr. Zulfikar Ali is the professor of MNS University of Agriculture, Multan, with research expertise in plant breeding and genetics. Currently, he is working in the areas of resource conservation, climate smart breeding. Okay, so let's have Professor Ali to share with us climate smart agriculture research at Multan. Please.
Thank you, Professor Lam. Am I audible?
Yes, we can see.
Yeah. So greetings to all participants from MNS University of Agriculture, Multan. In my presentation, I will just briefly go to the climate smart agriculture research we are doing here at the university for new seeds and agronomy of crops for the fruits, particularly mango, vegetables, some machines, and other some miscellaneous activities we are doing. So in agriculture, what we all know is very important at this university with the help of University of California, Davis, and the funding from USAID and Punjab Agriculture Research Board. We have modified the wheat architecture for the novel leaf traits to capture the gaseous water, converting to drops and moving to the root zone. And this wheat is basically self-irrigating and saving 30 to 50% flood irrigations compared to the varieties at the moment we are cultivating in Pakistan. This wheat has completed almost the VCU and DUS test. This is the last year. I'm very much hopeful that this year this wheat will be approved for the cultivation of by the farmers. To overcome the heat problem in wheat, we are increasing the phenotypic plasticity to absorb at least 5 degrees Celsius fluctuation on both extremes and to have 10 to 13% yield gain as well. Another work is very innovative with the collaboration of University of Sydney and the European seed company KWS. Funding is coming from the Horizon 2020, and in this project, we used BLA technology for the male sterility and maintenance, and developed more than 1,000 wheat hybrids in Pakistan, and also evaluated at more than 20 locations. And we have identified around 8 hybrids which are producing more than 20% above the cultivars. These hybrids, they are disease resistant compared to the OPV parent variety at least 1 week early compared to the parent and the varieties. National Assembly of Pakistan recognized this work of the university and issued the appreciation for this work. And topper hybrid from this work is has been submitted for the VCO and DUS testing this year. To improve the soil health and organic matter and water holding capacity, we are working fertilizer coated biochar, and it has very promising results, almost 2 to 3 times higher yield in cotton. With the Chinese Academy of Agricultural Sciences, the BRIE Professor Zhang Rui, we are working on the genetic adaptability and water fertilizer regulations. We are exploring the 30 years data of cotton belt in Pakistan and China in Xinjiang, and we will plan for the next 50 years. How should we develop the varieties and bring the gene packages to address the climate change in future. With the University of Kassel and funding from DAD, we are working on the biodiversity project where we are looking for the neglected and underutilized species diversity in the lowland of Cholistan, which is the desert, and highland of Gilgit-Baltistan. So this is the first year of this project, and in this project, we are building capacity with the exchange of faculty and students with the University of Kassel. For the climate, smart crops, and to diversify the diet and to diversify the crops to mitigate the climate change effects, we are working on the minor cereals, and particularly the quinoa, proso millet, and we are also introducing soybean in Pakistan. In the fruits, we have introduced some multi system in mango, guava, and citrus. And in mango, we have trained more than 400 mango growers, and out of them, more than 50 growers we took to Egypt to see the multi system orchard over there, and this is a trainer, Steve from South Africa. We brought him here to train the farmers, and up till now, more than 8,000 acres of small tree system of mango has been established in Punjab and Sindh. We we the production of biofortified pesticide-free vegetables in the hydroponic, where we are demonstrating different types of tomatoes and bell peppers. We have fabricated a machine, the load machine, with the cooperation of Australia and the local research institute. This machine basically drill cereals, pulses, and oil seeds with the low tillage, so save energy. It also place the seed at uniform depth and have good crop stand and reduce the seed rate. We have fabricated another machine which pull the cotton plant and shred it, incorporate into the soil, and this also crush the bolls where the major pest, pink bollworm, is sitting, and it is one of the control to control this pink worm. And it also has a weed sowing drill, so only single pass run. It will cultivate the field, pull up the cotton into the soil, and sow the wheat. We are also working some microbial work, particularly on bacteria and fungi, specifically pH reducing bacteria, phosphorus mobilizing fungus, and some drought tolerant fungus in the form of seed biotechnology. We are doing some pilot testing. We have demonstrated on-farm water harvesting in the saline areas where we are demonstrating the shrimp farming and fish farming. We have Plant for Life Society, which is focusing the plantation of trees to mitigate the climate change effect. Uh, uh, we are #1 in Pakistani universities and 154 in the global universities in the UIG Green Matrix, which indicates the sustainable actions of our campus community. We also hold climate smart conferences on the climate smart agriculture with the international collaboration, particularly with the University of Germany, and we are holding this conference annually. These are the international and national partners which are working with us in different types of research related to climate change. And I acknowledge their support, and I look forward in the next presentation after this today's activities. Chinese University of Hong Kong logo will be sitting in this slide. So thank you very much.
Thank you very much. I think it's very interesting and cover lots of areas. So the next speaker is from our side, from the Hong Kong side. Professor Amos Tai is a member of our state laboratory and is an associate professor in the Earth System Science program at our university. His research is very much different from mine. He aims to address the pressing issue of air pollution, climate change, food insecurity, and forest degradation using Earth system modeling and analysis of global observations. Now let's invite Professor Tai to share with us on modeling food, agriculture, environment interactions to help meditate air pollution and climate change. Amos, please.
All right, thank you very much, Hongming, for the very nice introduction. So as Hongming has mentioned, my research has focused mostly on interactions between pollution, climate change, and natural vegetation, as well as agriculture. So about half of my work is actually more about how forests would actually be used to help mitigate climate change and air pollution, things like that. But I will today, of course, focus on the part of my research that is on agriculture. So in particular, I have a strong focus on the impacts of air pollution and climate change together on agricultural productivity, which I'm going to explain soon. On the other hand, we also very interested in seeing how more sustainable farming methods as well as food consumption pattern can help mitigate pollution and climate change. So I will focus on these two parts today in the next 9 minutes or so. So as Hongming has mentioned, my methodology is mostly Earth system modeling integrated with a statistical model, a statistical analysis. So by Earth system modeling, basically, we are talking about process-based modeling, really using supercomputers to solve the thousands of equations that we know of to describe physical processes, chemical processes, and biological processes in the Earth system. And we put all of these equations in the computer, trying to solve them, dividing the whole world into many different grid cells, and also using integrating satellite observations and other sorts of observations as well into the model, so that the models themselves are not entirely fiction. They are actually based on reality with the observations as input and also using observations to validate our model. So I'm not going to talk too much about the model validation for now, and instead I'll discuss a very important issue. First part of my work is about surface ozone together with climate change, because this is a very important air pollutant that have tremendous damage on plants. So ozone, we often say it is good up high, bad nearby. The ozone that is up there in the stratosphere is usually good for us because they screen away the UV radiation. But if ozone exists around us near the surface, then they're really bad. They can damage the plants and they can damage our human health as well. As you can see, as we breathe in these ozone, then our lungs would be really, really bad. And they would also disrupt plant tissues like this. And thus, actually, a lot of studies have also shown that ozone greatly damage crop yield at ambient range of ozone, and indeed at Pakistan, India, and many parts of East Asia and South Asia suffer from this range of ambient ozone. And under this range of ozone, crop yield can be reduced by up to about 20%, reduced by up to 20%. So in some of our work, we try to see how air pollution, mostly ozone, together with climate change can combine to affect agriculture in different parts of the world. And we have to look at the co-effects of climate change and air pollution, because climate and atmospheric chemistry also interact with each other. For instance, ozone is also known to increase with temperature. So in a work that we have published a couple of years ago in Nature Climate Change, we have seen that the combined effects of ozone and warming are larger than their individual effects, because they kind of worsen each other and exacerbate each other. And that warming and ozone pollution can cause a global crop production loss of up to about 15%. And actually in South Asia, ozone can cause wheat reduction, maize reduction, and soybean reduction. For wheat, it would be up to about 20 to 30%, 40% indeed in the future scenario. So we really want to emphasize how air pollution control can have substantial co-benefits for both human health and food security. So through modeling and prediction using future climate and air quality projections, we are able to kind of assess the risk of different crops and in different places to air pollution and climate change together. That is one of the things that we are focusing on right now. So we try to use different metrics and see what kind of crops and in what kind of places, including South Asia, would be most susceptible and vulnerable to future changes in climate and pollution. So that is, I would say, half of my research related to agriculture. And in another half, we want to kind of see the other way around. We want to see if we can improve agricultural methods and food consumption patterns in order to reduce pollution and reduce greenhouse gas emission. So that is the other way around, looking at the other side of the cycle or arrows. So in one of the work that we have done recently, as Honming has already mentioned, intercropping is a very important sustainable methods of farming that can have environmental benefits as well as yield benefits. So, but Honming has mostly focused on what benefit it can bring to the farmers and also to the surrounding and immediate environment. Indeed, we also wanted to see how intercropping can have even larger scale impacts on the entire, say, on a national level, for instance. So we want to see how. So, for instance, in this particular piece of work, what we have done is to make use of different sorts of a process-based model, and in the model, we divide sort of an experimental scenario where the entire China implemented intercropping. And then we modeled how ammonia emission would be reduced in when intercropping is implemented across the entire China, all right? And then we use chemical transport model to see the subsequent impact on air pollution. So what we have found is that in organic PM2.5, particulate matter can actually be reduced by quite a bit if the entire Chinese agriculture is converted to soybean intercropping, all right? Because ammonia emission can be reduced upon lesser use of fertilizer if we implement intercropping. And thus, we also show that because we save fertilizer, we also reduce air pollution from ammonia emission and the PM 2.5. So there's also a saved health cost there as well, which is up to 13 billion US dollars per year. And finally, we also want to see how the changing Chinese population, which also, as well as nationwide conversion of a plant-based diet to a meat-intensive diet would affect air pollution and public health. So many people already know that eating more meat would have direct health consequences for the people who actually eat the meat, like, you know, diabetes, cardiovascular diseases, or cancers. Eating more meat would also emit more greenhouse gas emission, which cause climate change. These are something that everybody knows, everybody knows. But our research has further shown that eating more meat can also enhance ammonia emission, which is not a greenhouse gas, but it would contribute to particulate matter air pollution, and thus, which is a very bad, you know, air pollutant that would indirectly worsen health, causing respiratory diseases. So in this paper we just published in Nature Food, we have found that, actually over the past, I should actually show this one. Over the past 30 years, PM 2.5 has increased a lot in China due to a variety of reasons. And we have shown that up to 20% of the increase in PM 2.5, up to 20%, is actually arising from heavier meat consumption of the Chinese population. Another 80% would be from, you know, cars and industries and all of those production, energy sector, things like that. But we have shown that up to about 20% of the PM 2.5 increase over the past 30 years is indeed driven by more meat production. So we basically want to highlight how, and on the other hand, if we move back to a more sustainable plant-based diet, as recommended by the Chinese dietary guideline, we can reverse the result, reverse the situation. We can reduce ammonia emission, reduce particulate matter pollution, and reduce premature deaths related to particulate matter. So to conclude, we see that having a healthier diet, addressing food consumption can also be a very important way to reduce agricultural pollution and thus the subsequent health effects on population. And that would be applicable to India, to Pakistan, and the rest of South Asia as well. And we also show that efforts to implement sustainable farming methods like intercropping can also have substantial co-benefits for both the environment and human health and food security. So that is basically, finally, just allow me, because I've seen that many people here are working on water saving technologies for agriculture. We have also tried to see if water saving irrigation in China, like drip irrigation, or versus more water consuming irrigation methods like flooded irrigation or sprinkler irrigation, how these different types of irrigation would affect local air quality and climate, because the availability of water, basically you're adding a lot of water to the land surface, and that can change climate and change the atmosphere. So we see that actually the effects of flooded irrigation is a lot. Flooded irrigation can indeed reduce surface temperature because you are really adding a lot of water to the land surface. And thus, it can also reduce convection and trap more air pollutants in the boundary layer, leading to actually higher ozone and other air pollutant concentration. So, and what we've seen is that drip irrigation actually have a minimal impacts on climate and air quality, and thus would be a preferable way of doing irrigation in especially these semi-arid regions. So with that, I would just like to close, and if you have any questions, please feel free to let me know.
Thank you, Amos. So Amos has presented a kind of different kinds of talks, right? Using the big data and modeling. So he wants to address some global issues related to agriculture that will link to air quality. I think that is quite interesting. And he also advocated for less meat diets to protect our environment. So we could discuss it for further details in the discussion session. So next speaker is from the Pakistan side. So Dr. Aman Habib is the scientific officer of Agricultural Biotechnology Research Institute of Agricultural Research Institute, Faisalabad. His area of expertise are genetic modification of wheat and cotton plants for developing tolerance to drought, salinity stresses, and Roundup herbicide. Let's invite Dr. Habib to share with us on impact of climate change on local agriculture and roles of agrobiotechnology. Please.
Thank you, Dr. Lam, for a nice introduction. And greetings to all the participants from Chinese University and Pakistan. First of all, the topic of my today's talk is very important, and it is the impact of climate change on local agriculture and role of agri-biotechnology. First of all, I want to introduce that what is Pakistan's geography and how and what is climate. Pakistan is a blend of coastal areas, vast plains, and very high mountains, as you all know. And its area is 800,000 square kilometers. Now, 2/3 of Pakistan is lying in the arid zone. And we are having continental climate with very extreme temperature variations in summers and winters. And for example, in winter, we can get -20 degrees centigrade in northern areas like Skardu, whereas in summer months, like in southern Pakistan, we can get 53 degrees centigrade. So there is a very wild climate change in upper and South Pakistan. So this is the preview of the type of climate we are having already. Now, the climate change scenario that Pakistan is facing, I just want to introduce to that. Pakistan ranked 5th most vulnerable to climate change in the Global Climate Rank Index in 2020. So, and we have already lost 9,989 lives, according to that report, due to the different climate change, weather patterns, calamities, etc. Now, Pakistan has faced economic losses of about 3.8 billion since 1999 due to climate change, and witnessed 152 extreme climate events. That include floods, landslides, droughts, heat waves, and blizzards. Now, how this climate change is impacting our local agriculture? First of all, I want to focus on its impact on crops. Now, wheat is the most important and our staple food. So first of all, I will talk about wheat. We have seen that the crop growing season of wheat is shrinking. It is getting less time for grain filling and maturity. So as our scientists have also reported that the sowing time of wheat has shrunk to about 20 days. We sow wheat in 1st November a few years back, but now its date is 20th November that our agriculture department is recommending. So similarly, in the wheat season, we are seeing erratic rainfall patterns. Less rainfall is falling on vegetative state and more on maturity phase. So this is ruining the yield of wheat. Similarly, in the middle of the wheat season, we are facing sub-zero temperature, which is affecting the anthesis phase of the wheat and causing sterility and ultimately the yield losses. On the other hand, we are facing prolonged period of drought in the at sowing stage. So this is also reducing the germination of the wheat, especially in our rain-fed areas of Pakistan that only depend on the rainfall. And there is also a problem of terminal heat stress in the wheat, especially at the terminal stages. It is also affecting the grain filling and ultimately reducing the grain weight of wheat. And then in cotton, we are also facing the high temperature that is causing the flower and bole shedding in cotton, and ultimately we are losing a yield of the cotton. So this is a little bit of preview that how the climate change is impacting Pakistan. So first of all, its impact on the agricultural land is also very visible. Pakistan has cultivated land of 23.8 million hectares, which is about 30% of the total land mass. And it is getting 0.04 million hectares getting saline each and every day due to the climate change scenarios. Similarly, 15 million hectare has already been abandoned due to the drought stress that we are facing year on year. Pakistan has lost about $2 billion to drought stress, $1.5 million due to waterlogging, and about 300 million per annum due to various stresses. Now, how this climate change is impacting the insect pest complex that our crops are facing. So change in temperature and precipitation will change definitely the feeding habits, movement and reproduction patterns of insect pests. And we are already viewing this on our crops. Insects are shifting to non-host crops. For example, armyworm was a big problem in a decade ago on wheat, cotton crop, but now it has shifted to maize crop due to climate shifting. So the pink bollworm attack has sharply increased on cotton crop. Whitefly numbers have dramatically increased. So this is a virus vector. So virus transmission has also doubled in cotton crops. And we are facing wet and humid weather in cotton. So aphid is also on the increase in wheat and cotton. And after many decades, record locust swarms have damaged record crops in Pakistan. This is also due to the climate change. Similarly, climate change is also affecting the pattern of crop diseases. Prevalence of one pathogen over the other is occurring, especially in wheat. A few years ago, brown rust was the main problem in wheat during season, and now it has totally been shifted to yellow rust. So we are facing the yellow rust problem now instead of brown rust. And due to more humidity and rainfall, especially in the months of April and May, we are facing more viral diseases in cotton and vegetable and oilseeds. Due to the population load has increased of different viral vectors like whitefly, aphid, and JCIDs. Now, weeds are also showing the impact of climate change, and they are also modifying their habitat. And we are seeing that weeds they harbor insect pests during the off season, and then when the crop is in the field, those insect pests shift to our crops. So estimated 40% yield losses are reported in Pakistan due to these weeds. And we all know that most of the major weeds are C4. So the photosynthetic pathway. So C4 weeds can flourish in higher or rich CO2 levels that we are seeing in the climate change scenario. So models are also predicting that the changing in wheat biology, ecology, can interface and in the wake of climate change. So all these, after telling you all this scenario that what we are facing in Pakistan, now how can the biotechnology in this whole scenario can help us? So first of all, there are different approaches in biotechnology, and most important is the genetic engineering approach that many researchers are now doing in all around the world. And many of the genes and hundreds of genes have been isolated, transformed, characterized in different types of crops. So these are related to heat tolerance, drought tolerance genes, salinity tolerance genes, cold tolerance, related to different diseases, insecticidal genes like CRY1, AC, CRY2AB, WIP3, and herbicide genes, BAR, EPSPS, and many others. So overall, these genes have been transformed in many crops. Events have been characterized, like this table is showing that for abiotic stresses, 11 genes have been transformed. For disease resistance, 342, and for herbicide, 294 genes have been transformed, characterized, and these crops are commercialized in the world. For genomic approaches, there are now different genomic approaches that scientists are using that can help us. For example, genome editing approaches are now quite in these days. RNA-seq techniques that can identify the functional gene related to abiotic and biotic stresses. Then there are molecular markers available, SSR, ISSRs, through which we can screen our material available germplasm that is available to our crop institutes. We can screen it for drought and different stresses genes. Then we can associate those genes with association mapping. Different traits can be associated with the markers, and then those markers can be used for various stresses in the future. Tissue culture approaches can also help us. So, for example, somaclonal variation, drought, heat, stress, we can screen. We can create plants with somaclonal variation, then check these plants for heat, stress, cold, and drought. Then micropropagation can be used and is being used for creating disease-free seeds. And we can optimize calogenesis, organogenesis of our GMO crops for developing new events. And when we can create variation through different mutants like EMS, we can use that to create mutations in our materials. Now, what my institute, Agriculture Biotechnology Research Institute, is doing in this scenario, we are first of all doing genetic engineering against different type of insects in cotton, in wheat, in brassica, and sugarcane. We have optimized the codons and then synthesized the different genes like EPSPS for herbicidal genes, for CRY1, AC, CRY2AB for insects, chewing insects. And we have already transformed them in these 4 crops with the help of agrobacterium mediated transformation. And the insecticidal genes, these are transformed in tobacco, and we are in the stage of characterizing these events. Similarly, we are using different genomics techniques, like we are validating different molecular markers against drought stress and also against wheat rust. And we have found many new markers against the prevalence rates of these diseases and insects in our material. Also, we are using tissue culture approaches to create variation in already available materials that different crop institutes they share with us, and then we create a variation in this material through somaclonal variation and micropropagation. And this institute has released two varieties, one of wheat and one of sugarcane. And our material also, we take part in national and provincial yield trials. Similarly, we are using different, as I told you, mutagenesis like EMS, ethyl methyl sulfonate, to create variation in the genome and screen that for different types of stresses. For example, this experiment is related to screening of sugarcane germplasm against the stress, drought stress. And we have found quite a few plants that we are checking for further for strep tolerance in field. This experiment is related to sugarcane and star salt tolerance. We are like supplementing the nutrient media with various level of salinity and then screening the sugarcane material. Fourthly, we are also using the crop modeling like DSET and Epsom for making ease in the decision making for in the climate change scenario. So one of experiment that we have done is to predict the impact of climate change on wheat crop by using crop modeling to better prepare varieties to minimize the yield losses. We have used these two wheat varieties and created two climate change scenarios. In one, we have created 9 treatments. In second scenario, we have tested 6 different type of climate change scenarios in which we have changed the carbon dioxide level, temperature, and rainfall. So after the crop modeling, the model has told us that the CO2 level, if it is 396, that is the current level, what will be the impact of when we will check it on the higher CO2 levels and higher rainfall and higher temperature. So the crop model has told us that only 1 degree increase in the temperature could cause 20% yield losses in our wheat varieties. And if we increase the temperature in combination with the higher rainfall, that is 25% higher rainfall, that can cause even more losses, that is 30.8%. So in the future, with the higher level of temperature, we may also see higher level of rainfall. So both of these stresses, when they combine, they will cause more yield losses. So we have to prepare ourselves for this scenario too. So I want to conclude my talk and Pakistan is a developing country, and like Pakistan, which is the most vulnerable to the climate change. So we need to ensure our food security and sustainable crop yields. Similarly, we need to adopt state-of-the-art biotechnological approaches that the world is using, like RNA-seq. We got hundreds of germplasm that is available to our crop institute at AoE Agriculture Research Institute. So we can screen that material through RNA-seq for the functional abiotic and biotic tolerant genes. And then we can use these genes to breed new varieties that are better performing against climate change. Similarly, genetic engineering can be used to transform those the genes that we have identified through RNA-seq into our varieties. New varieties can be created through genetic engineering. And lastly, genomic editing approach that is a new approach. We can also use that approach to improve the genetic architecture of stress susceptible crop varieties. Thank you.
Thank you, Dr. So I believe that both using the genome editing, transgenic approach, and make use of the original germplasm are all possible approach to improve the seeds, which will be important as important as improving the agronomy practice. So far we have covered different things: agronomy practice, approach for seeds, climate modeling. Now, so before the first break, I would like to introduce my colleagues who will talk about the nutrition, because now so far we're talking about how to grow the plant, but after growing it, how could it be a good nutritional value to human consumption? So Professor Jack Wing-Wong is one member of our State Lab and an assistant professor of School of Life Sciences at the Chinese University of Hong Kong. His main research areas are on vascular and metabolic biology, stem cell biology, and cardiovascular regeneration. However, I had invited him to join our agricultural team. Today, Professor Wong will share with us on the topic short-chain fatty acids and cardiometabolic syndrome. Jack, please.
Thank you, Professor Lam, for your nice introduction. Good afternoon, everyone. It's my great honor and pleasure to meet so many esteemed agricultural scientists from Pakistan at one time. So nice, very nice to meet all of you. And I enjoyed so much. Actually, I myself is not, as mentioned by Professor Lam, I myself is not an agricultural scientist, but I enjoyed a lot from the talks just now, and I also look forward to the more exciting and interesting talk coming forward. So as mentioned, my name is Jack Wong. I'm an assistant professor at the School of Life Sciences. So today, actually, my aim is, as mentioned, I'm not an agricultural scientist. Instead, I'm actually a physiologist, trying to understand the mechanisms of how our body work. But I believe there is some reasons for me to join this group, because I think, as mentioned from Professor Lam, all these actually there are so many bioactive compounds that can be found from the crops. I believe that there are some ways for us to collaborate to understand more about the beneficial effect of the bioactive compounds related to our human health. In this regard, actually, let me introduce in this 7 or 9 minutes talk, introduce what my group is actually doing. So as mentioned, the main research areas in my group are focused on vascular and metabolic biology. We explore the local mechanisms that contribute to the endothelial dysfunction, the vascular dysfunction. Endothelial cell is one of the innermost lining of the blood vessels that cover all inside our blood vessels, all inside our body. So we also actually focus on the stem cell biology. We use the iPS cells, induced pluripotent stem cells, to understand the disease mechanism, to do the disease modeling. And furthermore, we also focus on the cardiovascular regeneration. Particularly, we want to illustrate the crosstalk between immunity and the cardiovascular biology. So this is the mission statement of my group, one group. So we aim to define local molecular mechanisms in stem cell biology and vascular biology, which are essential to develop diagnostic and therapeutic strategies for treating and managing widespread conditions. So we actually indeed work with many clinicians, many scientists locally and internationally. And we also work with physicians to keep our basic findings, basic research findings can also be translated to the clinics. So this is our mission statement. So today is what I'm planning to do is to share you a few of our recent work published actually this year or the last year. I will just share three of them. So you can actually see, if you're interested in our work, you can just go online and throughout these 10 or 15 years or so, we have been focusing our research effort in publishing work related to the vascular and metabolic biology, stem cell biology, and the cardiovascular regeneration before. So today I'm going to share three stories briefly. One is about our immune-mediated revascularization. Second is the iPS cell disease as a model, induced pluripotent stem cell as a model for disease modeling. And also, we also try to, at last, we actually focus back on today's today's focus on the short-chain fatty acid and cardiometabolic syndrome. But I will just go briefly over it because of the limitation of time. Instead, I will actually give you the, if you are interested, I can give you the print for the papers to read. Okay, so the first topic is on immune-mediated cardiovascular regeneration. This work actually is done mainly by my PhD student, Mr. Francis Chen. So he just got his PhD degree from my team. And in his work, actually what happened is, I will just give you a highlight of his research. So what actually happened is, we all know that for the cardiovascular region, for the patients that have the myocardial infarction, the heart attack, the adult heart itself cannot regenerate. But interestingly, but if there is injury or ischemic injury in the neonatal, in the newborn baby, the heart can regenerate. And that's actually holds true with the lower end animals like the silverfish or the axolotls that can regenerate pretty good. And our recent findings suggested that, okay, in the adult mice, although they cannot, in the adult mouse model, we cannot, the heart cannot regenerate after the left anterior descending artery ligation or the coronary artery ligation. But the neonatal, but the newborn mice at the, you can see from the 2 day per post injury. So at the 2 days after birth, we ligate the coronary artery. So in the control mice, you can see this is the percentage ejection fraction, the heart function, they can restore to normal. On the other hand, if this is a DKO mice, in this DKO mice, there is no interleukin-4 and interleukin-13. This is a double knockout mice. In these mice, they do not express the top 2 cytokines, the interleukin-4 and interleukin-13. In this work, to just sum up very briefly, the highlight for his work, of Francis' work, is that he demonstrated that the interleukin-4 and interleukin-13 are necessary for neonatal cardiac regeneration. And in this case, he used multiple models, including the double knockout transgenic mice and myeloid cell specific interleukin-4 receptor alpha deficient mice. He demonstrated the importance of the top 2 innate immunity in cardiovascular regeneration. So I will just give this story very brief. And if you're interested in this work, this paper has just been published in Theranostic and entitled "The Top 2 Innate Immunity Drive the Distinct Neonatal Immune Profile Conducive for Heart Regeneration." In this regard, actually, we have all these advanced technology and tool for probing on the in the mouse immune function. So you can imagine if you have a bioactive crops, or if you have interested bioactive compounds, for example, from the crops, we can test it easily because we have all these immune panels on the animals models that we can test it on the regeneration. Okay, let's put it this way. Okay, this is the first story. So in another work, as mentioned from the stem cell biology, this is another work from my another PhD student, Vimo Guru, and he is in his last year of the PhD study now. And what he has done in this case, he has tried to collect the blood samples or some skin samples from real patient. And what this advanced technique has been doing is he generated the induced stem cell from the somatic cells, and then try to differentiate these induced pluripotent stem cell into the cardiomyocytes and study their function. So basically, the highlight for this talk is he can use this model, this iPSC-derived cardiomyocyte, to study the channel diseases related arrhythmia. And in this case, again, we can use this model to do the drug screening. And actually, we can extend it forward. If we have a panel of the bioactive compounds from the crops or from the food, we can use this model, this cell-based model, to understand the effect and the mechanisms of the beneficial effect of some bioactive compounds on a cell-based, on a human cell model, which it is actually very hard. For example, it's very hard to ask somebody to give you a heart to study, but using this model, we can produce the stem cell, induced pluripotent stem cell, and then we can differentiate them into the cell type we want. In this study, we use the cardiomyocyte, but we can also differentiate these iPS cells into endothelial cells or into the neurons. So again, to cut the story short, if you feel interested, you can also look at this publication we actually just published in November last year. So this is a stem cell-based model that we use to produce the cardiomyocytes. And apart from the cardiomyocytes, we can also produce the blood vessel cells, the endothelial cells, or the neurons. Actually, this may help to understand how these different bioactive compounds from the crops may be beneficial to our human health. Okay, so finally, my third sharing is on my PhD. Actually, this is already graduated a PhD student, Qin Tian. Dr. Tian actually she investigated the effect of some this type of the short-chain fatty acid, the butyrate or the butyric acid on the endothelial function, and by using the transgenic model, she proposed that the butyrate may act on a nuclear receptor, the peroxisome proliferator activated receptor delta, PIPA delta, which may increase one of the microRNA expression and reduce the reactive oxygen species level and promote the vascular health. So again, this work basically demonstrates to you that we have the animal models, we have the cell models to understand or actually to investigate the protective effect of some bioactive compounds that may be beneficial to our health, particularly the cardiovascular health. So again, if you feel interested, you can just take a look at this paper published in Pharmacological Research, published I think in July just last year. Okay, so anyway, so here, as I'm not an agricultural scientist, but I'm actually do feel very interested in Professor Lam's work and as well as your work. And I think I will just do my work, introduce myself to you. And if in case later on or in if you have any interest in understanding the effect of some bioactive molecules, bioactive compounds from the crops, feel free to contact me. And our strength rely on, just show here, we have both the cell-based model, animal models to study the metabolic syndrome and rare diseases, and we have all these tools to understand the mechanisms of the diseases, particularly on the cardiovascular diseases. We have a broad focus in understanding the pharmacological effect of some herbs, particularly on the traditional Chinese medicine and the food supplement. We also have close collaboration with clinicians and basic scientists, and also we're interested in doing those translational research that actually can benefit to patients, to real patients. So with that, I would like to stop here. Thank you all for your listening. And this is my research group. And yeah, and I think I will end here. I will just end here. Thank you, Professor Lam. Thank you all.
Thank you, Jack. So for agricultural products, so we are always looking for how to increase the income of such product by value adding. So making the product into nutraceuticals is one way to become nutritional supplements. For example, soybean can make into isoflavones and lecithins. Those are much more economic value than just raw seeds. So Jack's expertise would be very helpful if any one of us has a underused crops that might have some nutraceutical values. Then we can encourage the farmer to go for value-added products. So I guess we have finished the first session. In order to give everybody some time to drink a coffee, so I will call for a 10-minute break. Hi everyone, I think it's time to start our session. I guess everyone hopefully is ready after a cup of coffee. So the first speaker of the second session is from the Pakistan side. So Professor Dr. Ghulam Murtaza is the director of Institute Soil and Environmental Sciences at the University of Agriculture, Faisalabad. His research interests include soil salinity, salticity, reclamation of salt-affected soils, nitrogen use efficiencies in normal and salt-affected soils. Let's invite Professor Murtaza to share with us on agriculture, land degradation, and environment concerns and solutions. Please.
Thank you. Thank you for providing opportunity to present on land degradation and environment consequences and solutions. Thanks for nice introduction as well. As I have selected a topic for land degradation, and you know that soil is the medium which provides nutrition to, which acts as a media for plant growth, and it's a foundation of food production and supports ecosystem, and acts as a sink for storing and supplying clean water, and then help out for maintaining biodiversity and sequestration, carbon sequestration, and increase resilience against changing climate. Land degradation in relation to climate change, and you know that there are 5 major you can say drivers for changing the demography of the land. The major issues are of ever increasing population, and due to overpopulation, there are many other associated problems. More and more solid waste is generated, sewerage problems, and you know that due to anthropogenic activities, due to more and more industrialization, then there are gaseous emissions which are directly affecting the climate. These emissions also affect ozone depletion, and this change in the climate that's disturbing the biodiversity, flora and fauna. We are contaminating our freshwater resources, and secondly, due to overuse of fresh water for domestic purposes and for industrialization, the proportion of fresh water for crops is decreasing day by day, and all these ultimately affecting you can say the ecosystem, degrading the ecosystem. I mean, the soil is drastically, you can say, contaminated, and due to land degradation, 5 major drivers of land degradation are desertification, and then deforestation, and salinization, and compaction, and soil erosion. At global level, the total area which is declining soil productivity during the past more than 30 years is around 30%, and over 10 million hectares of arable land globally has been degraded every year on area roughly 1/3 of the size of the Germany. And this map shows red color shows very highly severe problem, and then this color yellow is showing high severity, and this brick color is showing moderate severity, and green color is and then worldwide about 52% of the land used for agriculture is moderately or severely affected by land degradation, and then 75 billion tons of fertile soil is disappearing every year. It's alarming for everyone that huge amount of you can say area of fertile soil we are losing, and about 12 million hectare per year is lost due to drought and desertification, and causing 27,000 species they are disappearing, you can say, just due to land degradation. As I earlier said that these are the main drivers of land degradation: overgrazing about 35%, and causing and deforestation is you can say the share of deforestation is 30%, and due to agricultural activities 28%, and over exploitation for biofuels 7%. And you know that centuries are needed to develop top few few centimeter top productive layer of soil to form. It may take centuries, but we are destroying that fertile top layer of soil. Just if we this slide, I'm showing the data of Pakistan. About 1 billion tons of top fertile soil is eroded every year, and due to which we are losing nutrients and depletion of soil fertility, and ultimately less productivity of crops. And another issue is soil salinization, and out of the total cultivated area, which is about 23 million hectare, almost more than one third of the area is salt affected due to higher concentration of soluble salts, or due to high concentration, high amount of exchangeable sodium, or both soluble salts as well as exchangeable sodium. And then desertification, especially in southern Pakistan, overstocking, intensive farming, removal of vegetation, forests, and due to other socio-economic and political reasons, lot of you can say desertification, and then pollution. I mean, as I said, that a lot of sewage water is created, and from industries and from municipal you can say areas, and unfortunately all is being mixed. There is no separate arrangement for the disposal of waste that is generated from residential areas, and that is you can say released from industries. Even hospitals water is being mixed in the same system, and unfortunately very limited at limited level. Just there is treatment of that effluents at primary level, mainly at primary level, and then there is physical degradation, I mean structural deterioration, compaction of land, and more labor and energy, less economic, and then unfortunately due to arid, semi-arid type of climate, due to high temperature, organic matter level is very low, that is less than 1%, and we need to another reason, that is improper land use. I mean, in those areas, just as one of my colleagues stated that, for example, in southern Punjab, that was considered for cotton belt, and lot of area has been replaced with maize and sugarcane. So improper land use, that is also you can say considered major problem, reason for land degradation. Here I have shown the data about how much area is affected by water erosion and wind erosion and declining soil fertility, and about 2 million hectare area is affected by waterlogging and salinization, lowering of water table. Here I have shown that huge, you can say, economic losses in billions, they are occurring just due to these issues. And you see that wind erosion contributes about 40%, 42% of agricultural land. So I will say that huge, you can say, potential for using this area for agricultural production. Here in this, I have shown the economic cost of land degradation, salt-induced land degradation in irrigated areas of the world. I'm just presenting this line is economic losses real value calculated on the basis of 1990 and then 2002 and then 2013. And this graph shows the economical losses, nominal value. Nominal here I mean that the changes refers to economic losses based on the basis of you can say the loss real values have been translated into the equivalent amount in 2002 and 2013 by adding inflation. Here we have collected data for major 4 crops in Pakistan and compare the yield losses just due to salinization, due to salt affected land. We have collected the data of normal lands and from the salt affected adjacent lands. I mean, just we have calculated yield losses from salt affected soils compared to their counterpart normal soils, and in both the cases inputs are same type of texture of the soil, same climate, same amount of water, same quality of water. I mean, all the inputs were same, but just due to salt affected land, 45% yield losses have been reported in case of rice. Similarly for wheat, we have calculated that 40% yield losses are occurring due to salt affected lands. As I said that all the inputs were same, and similarly in case of cotton, huge losses. Although in case of cotton, the major issue is germination, and then cotton is relatively tolerant at vegetative stage, but at germination it is very, very sensitive. And similarly, in case of sugarcane, we have reported that 41% yield losses, they are occurring just due to, you can say, due to salt affected lands. Finally, I will just propose some suggestions that revision of the present land classification system for land zoning and land use maps is required. Then crops must be sown according to land productivity or capability classes. As I said that lot of productive lands in the peri-urban of cities, they have been converted to residential colonies. So those lands are not meant for residential colonies. So there should be planning for proper land use and allocation of land. Then house colony should be built on lands of B and C category within a maximum limit of 2 years. Crop residue management to increase organic matter level in soils, and then nutrient indexing of all agricultural land, and a cyber network for fertilizer application is required. Then we have to manage agronomic and engineering options and policy interventions for managing salinity and sodicity issues of Pakistan. And then we should recommend management and treatment options for those salt affected lands on soil test basis. And finally, capacity building programs may be arranged through combined efforts of academia and extension staff, which is very important. And then finally, legislation should be done to keep the farm size under a viable productive unit. Finally, I will conclude that we have limited soil and water resources, and we have to manage these limited soil and water resources because it's our moral duty that we should hand over these limited natural resources to our next generations in neat and clean farm. Thank you.
Thank you very much. So now we switch gear to soil problem. I think it will cause a lot of interest because the next speaker will talk on similar topic on soil, but a little bit different, but it's also on soil and the sustainability of soil usage. So the next speaker, Professor Martin CUHK-Pakistan, is a member of our state laboratories and is an associate professor in the Chinese University of Hong Kong. His research tackles different questions as related to mercury cycling in diverse environmental types and focus on vegetated landscapes, especially forests, streams and rivers, and wetlands. Let's invite Professor Cui to share with us on mercury pollution in the environment from forest to agriculture.
Okay, thank you, Professor Lam. Nice to see everyone here today. So, I just joined CUHK last year. So, and I want to talk about a little bit on related to my current research on mercury pollution in the environment. But today we'll be focusing on how mercury pollution, we can see it from the oceans to the forest and to the agriculture. So just a very little bit of background on mercury. So we have different forms of mercury in the environment. We have a gaseous form of mercury we call elemental mercury or mercury zero. So that's all in our air. And some of this elemental mercury could be oxidized to form the so-called inorganic mercury. So inorganic mercury, you can find it in the soil, in the water. And some of this inorganic mercury may be transformed to an organic form we call methylmercury. So typically transformed by bacteria. And this form of the mercury is the very toxic form of mercury and of our biggest concern. So you may have heard about Minamata Bay in Japan. The incident was from 1932 to 1968, almost over a span of 40 years. So what happened there is a small fishing village in southwestern Japan. So there was a long-term wastewater discharge from a factories containing high levels of methylmercury. The seafood in the bay, fish, for example, were contaminated with methylmercury, and people were consuming them without knowing it for decades. So from the government record, at least about 1,700 deaths and many suffered from long-term diseases. So this was a big problem, a big disaster for humans. But mercury problem is not just limited to Minamata Bay. High methylmercury in fish We've seen that all over the world. So contamination of methylmercury has been found in fish and seafood everywhere. For example, this shows a post in the United States, some water bodies to tell people do not consume the fish from this aquatic body because of high levels of mercury. And there are lots of government suggestions on how to reduce the consumption of fish in order to reduce the exposures to toxic methylmercury. And there are also mobile phone apps, so to tell you which sushi you should pick in order to reduce the exposures to mercury. So therefore, it seems like we have a lot of problems from the oceans because of mercury, but forest ecosystems have also a lot of mercury problems too. So why this would be possible? Because trees, they can take up the elemental mercury, the gaseous form of mercury, directly from the air through the stomata. So when the stomata open, mercury zero enter and then being oxidized to form the inorganic mercury. So therefore, when the tree grow over time, so more mercury has been observed in the leaves. So when the leaves drop, okay, so the mercury could pollute their soil, and some of that mercury could become the methylmercury. So therefore, forest ecosystems having more mercury pollution in general. So let's look at a global view on mercury emissions. So emissions meaning those emitted from anthropogenic activities. So this is from 2010, around 12 years ago. So you can see that a lot of high mercury emissions happen in our areas. Okay? So in East Asia as well as South Asia. But they bear of different sources. Here showing the coal combustions is the main drivers for these regions. Okay? So it accounts for around 25% of the worldwide mercury emissions. And then the rest of that is driven by small-scale gold mining activities. So it accounts for about 40% of mercury emissions. So China becoming the biggest global emitters of mercury. So from different industrial sectors. And you can see that from since the economic revolution. Okay, so it's basically from 1980 to 2012. You can see that the mercury emissions overall at least increased by 45 times. So therefore, there are now a global effort in reducing mercury emissions. So it's been cited by into so-called Minamata Convention, became effective in 2017. So then, why mercury is a concern for agriculture? So that's the main topic for today's webinar. So just give you a very quick overview on the early days in agriculture. So I found the paper in 1950, published in 1950. So if you look at this table, okay, it shows that the plant, the onion plant, okay, so if you did not treat with mercury, so almost half of them would die. But scientists found that if they treat increasing concentrations of mercury, so they found that the plants seem to do much better. Okay, so one of the reasons is that the fungal infection, so could be removed, could be inhibited by the presence of mercury, especially methylmercury. So therefore, there have been a bit of applications using mercury for so-called seed dressings. So this is a drum used for seed dressing. And basically the idea is to prevent the seed-borne diseases and also protect the germinating seeds from any secondary infections. Eventually, farmers found that it increased the crop yields. However, the high methylmercury coated on the surface of the seed actually have some risk. One of that is in Iraq in early 80s. So there has been large-scale poisoning caused by the consumption of methylmercury dressed seed in the country. So what's the problem of mercury in modern days in agriculture? So one of that has been seen in rice field. Okay, so rice field is interesting because you have inorganic mercury, and water saturated field will promote the bacterial production of methylmercury, and methylmercury has been demonstrated to be taken up and into the roots and translocated into the grain of the rice plants. Okay. So therefore, the plant itself absorbs the methylmercury directly from the soil. So, for the majority of coastal populations, we take up methylmercury mainly by eating fish. But in inland China, in this case study, showing that rice rather than the fish is the major pathway for methylmercury exposures. So in the estimations, up to 95 to 96% of the methylmercury in the human body is derived from eating rice. So therefore, it will be a concern for these inland populations. If they don't eat fish, they mainly eat rice. So rice could become a major pathway for methylmercury exposures. So when we look at, we don't have much information in Pakistan. So I try to find that. So one of that is showing some elevated air mercury levels in East Asia, mainly from China, as well as South Asia, mainly emitted from India. So it shows here the country of Pakistan could have high levels of mercury in the air. But since a lot of southern portion of the country is desert, for example, so therefore the mercury deposition could be lower because there's no trees to pull down the mercury in the air. So, but still there are lots of rooms to do more interesting research in these countries. So finally, as a quick summary, okay, so methylmercury continues to be a concern worldwide. And methylmercury definitely poses a threat for humans, whenever you eat fish, rice, or other food products. So thank you all. My email is here. If you have any questions, feel free to contact me. Yeah, thank you.
Okay, thank you. Talking on the pollution from the ocean to the soil to agricultural field. I'm pretty sure you will have lots of common interests with the previous speaker. So and then let's move on to the next speaker, Professor Dr. Sahir Ahmed is the director of the Office of Research, Innovation and Commercialization and the professor of Institute Soil and Environmental Sciences at the University of Agriculture, Thessaloniki. His research interests include microbial biosynthesis of plant growth regulating substances and plant micro interactions for improving crop yields. Let's invite Professor Sahir to share with us on soil management for changing climate. Please.
Thank you very much. First of all, I would like to appreciate and commend the Consulate General of Pakistan for cooperating and bringing the two labs and the agriculture universities in the country together, and also Dr. Anjam Ali, DG Extension, who coordinated in an excellent way. And today we are together to share our views. So I'm going to discuss soil management for changing climate. Before me, Dr. Ghulam Murtaza, he did discuss different soil issues, which have made my job comparatively easier. So these are the different functions that soil performs. It, of course, delivers ecosystem services that enable life on Earth. Due to the Green Revolution, agricultural productivity increased at the cost of many benefits and costs. And soil quality is actually reducing due to agricultural intensification, urbanization, unsustainable agricultural practices, jeopardizing our ability to maintain production and food for the future. We have about 60 years of topsoil left. Soil loss is up to 40 times the rate of formation. Dr. Ghulam Murtaza was giving the data from Pakistan. 30% less food will be produced against projected demand. For too long, the world has taken soils for granted, whereas soil is a natural resource that is not easily renewed. These are the impacts of climate change on agriculture. I don't want to go into the detail. Everybody knows what happens due to the climate change. Actually, being soil microbiologist, I'm trying to save time for my last portion of the talk, where I'll be focusing on microbiology. So these are the impacts of climate change on agriculture. These are the impacts of climate change in Pakistan, like increased variability of monsoon, upstream intrusion of saline waters in the Indus Delta, and increased floods and droughts, food insecurity, reduced agricultural productivity, reduction in capacity of natural reservoirs, more rapid recession of Hindu Kush Himalayan glaciers, and risk to mangroves and coral reefs. These are the strategies to manage soils in agriculture, in climate change phenomena. But my focus today is on microbiology, how microbiology can be helpful for that. So we can use that for abiotic stress tolerance. So one of the important phenomena that is taking place due to the climate change is the abiotic or biotic stresses. Those may be the stresses due to temperature, due to salinity, due to water deficit or excess, or due to nutrients. So abiotic stresses can be tolerated or can be controlled through microbial applications. Detoxification of heavy metals can be done through different approaches. Bioremediation can be a very good approach. And this is the particular area that is the biofertilizers and biopesticides, where we are working on. I will be discussing this in detail, and nutrient acquisition. So this is just to give you an idea. I have selected this picture just to give you the idea that how the microorganisms, they can be helpful through certain indirect or direct mechanisms for improving nutrition, through lowering of ethylene, or through the growth stimulation and plant protection, through enhanced resistance against diseases, decreased sodium uptake, reduced osmotic stress, reduced reactive oxygen species. These are the different mechanisms through which the microorganisms, they can enhance the plant growth. Just you can see, this is the soil environment, and these are different stresses which may be present: pathogen, flooding, temperature, heavy metals, drought, salinity. So if we inoculate with certain plant growth promoting rhizobacteria, they can reduce the stress induced impact. And due to the negative impact of these stresses, you can see the inhibited growth. But these microorganisms, through these mechanisms, they remove the stress effect or the deleterious effect of these stresses, and plant growth is increased. This is actually the area where we are focusing. And I believe listening to your talk, very first talk, even your interest on the soybean and many other crops, this is actually the crux of our work. We are the pioneers in Pakistan. We work on such microorganisms which have the ability of producing a specific enzyme which is called as ACC deaminase. You being the scientists know very well that this ACC deaminase is actually the precursor of ethylene. And ethylene is a plant hormone. And whenever there is any stress, ethylene production in the plant is increased. And as a result, the root growth is inhibited. So what we do is we isolate such microorganisms which have the ability of producing ACC deaminase. These ACC deaminase, actually this ACC, say for example, this is the PGPR that we have inoculated on the seed or the root. This ACC, due to concentration gradient, goes out of the root, and there this ACC in the rhizosphere changes to ammonia and alpha ketobutyrate. And that can be used by the microorganisms or the plants even. And as a result, the ethylene production in the plant is decreased. And when that ethylene production is decreased, as a result, the inhibitory effect of any stress on the crop plants is decreased. So that stress we have tested, it is very effective in case of nutritional stress. It is very effective in case of drought stress. It is very effective in case of salinity stress. And it is extremely effective in case of temperature stress. So we have such isolates which, when used for inoculating the seeds, they can improve the crop growth through eliminating the stress-induced ethylene production. So that can then result in mitigating, or you can say in coping with the stresses which are induced due to climate change. So this is using that approach. What we do actually here in at our university is that we isolate such microorganisms which have this ability. We screen them in our lab, then we test them under exenic conditions, test them under pot and field conditions, and finally standardize the studies. And ultimately, we have developed these 3 products. This is a bioherbicide. This is another story. Bioherbicide, and you would be interesting. Again, we are the pioneers in Pakistan. Dr. Anjum, it would be of interest for you that we have isolated such strains which are very effective for suppressing the weeds, but simultaneously improve the crop growth. This product we have developed for wheat crop, and we have tested it to be effective against 3 weeds, that is little seed canary grass, then wild oat, and broadleaf dock. Against these 3 weeds, it was very effective, and simultaneously it improved the wheat crop. And this is what I was telling, particularly when I was mentioning rhizobium. This is our innovation, that we have combined the rhizobium with such bacteria which have the ability of ACC deaminase. So ultimately, this improves the nodulation in the leguminous plants. You are working on soybean. So in case of soybean, we have tried to make a combination of bradyrhizobium with such PGPR and an auxin precursor L-tryptophan. And it gave marvelous results as far as the nodulation is concerned and as far as the crop growth is concerned. And this one, the Rhizogold Plus, this has a consortium of such free-living bacteria which have ACC deaminase ability, production ability in them. And this is very effective under stress conditions. So whenever there is salt stress, there will be more ethylene production. then these bacteria will be effective over there, and they will be reducing that stress-induced ethylene, and as a result, the crop growth will increase. So these two products where we are using the consortium. These are basically focused on ACC deaminase, and this one, the bioherbicide, in this case, we select such bacteria which these days are called as allelopathic bacteria, which have the ability of hydrogen cyanide production. Then they have the ability of, you can say, in case of E. coli, they have the anti-metabolite assay, and they have the lettuce seedling assay that is used for screening these. And ultimately, we have tested it for wheat, and these days we are working for using it for rice. And in addition to that, we are also working on a very interesting approach where we are using cow dung along with sulfur and sulfur oxidizing bacteria. And as a result, what we get is a compost which is of very low pH. And when that product is added, a sort of compost of low pH, when that is added to the soils which have high pH in our country, then it gives a sort of quick shock to the soil, and the nutrients are made available. And to further make it more effective, what we have done is we have one of the issue in our country is the dead poultry. We have big control sheds, so there are many dead poultry birds. So what we have done is we have dissolved those dead poultry birds into a specific chemical, potassium hydroxide. As a result, the product was of very high pH. We mixed that very high pH product with that low pH product. And as a result, we got a very nutritive fertilizer, which is particularly very important. Dr. Anjum Saab, particularly it is very important considering the current fertilizer crisis in our country. And even if we can combine this product with rock phosphate, this can be a very good source to supplement the phosphatic fertilizers. Because your lab is focusing on biotechnology, so as Director Auric, I would say that we do have one Center for Advanced Studies in Agriculture and Food Security. They are working on different aspects of gene editing. Then we have a Center for Agricultural Biochemistry and Biotechnology. So a lot of biotechnology work is going on at campus. So we will always like to have a more exhaustive and you can say detailed discussion with your lab to collaborate on any of the areas of the biotechnology, or I personally would like to collaborate on the microbial approaches for combating the impacts of climate change. And finally, I would also like to share that we do also have a Confucius Institute here at our campus. And I am heading that Confucius Institute here at our campus, which is, the Chinese Language and Culture Center. Thank you very much. And we will be discussing in the discussion session in detail.
Thank you very much. So I'm very much interested in the development of biofertilizers using microbes, and particularly you're interested in Confucius, so we have a lot of common things to talk about.
Yeah.
Okay, so we will talk more during the discussion, but let me introduce the next speaker, and he's doing something different, but also very important for agriculture and study of vegetation. So Professor Jin Wu is a member of our State Lab, and he is an assistant professor of School of Biological Science of the University of Hong Kong. He's from another university. His research interests include plant ecology, plant physiology, ecology, biodiversity, plant functional traits, ecosystem function, and multi-source remote sensing. And he's expert in high spectral analysis. Let's invite Professor Wu to share with us on solving climate change threats by integrating ecophysiology theory and big data. Professor Wu, please.
Thanks so much, Professor Lang. Can you hear me okay?
Yes. Okay.
Also, I really appreciate all the great talk. I really enjoy it. And like Professor Lang point out, I'm not an agricultural scientist, but I'm kind of want to become an agricultural scientist. So that's the reason I feel really excited. Today I have learned a lot from everyone. And today I bring to the table is my plant ecophysiology background and also remote sensing technique and potentially also ecophysiology modeling. So I hope that can provide at least some new insight and also can be potentially beneficial as well. So so here is a brief background of myself. I'm currently a PI of the Global Ecology and Remote Sensing Lab at the Hong Kong U. And my lab actually primarily is trying to address the global ecology issues actually. And with this, we divide our lab research into the 4 major research directions. One is called the plant ecophysiology mechanism. And the key things here is try to understand how plants respond to the different climate change stress. And secondly, we also focus 2 different like the new technology development. One is called a high throughput phenotyping technique. Originally, we developed this for vegetation modeling. Later on, now we found that this also quite important for the agriculture field. At the same time, we also try to leverage like a multi-scale remote sensing technique so that we can kind of like to integrate this kind of like ground-based observation to even larger spatial and temporal scales. And finally, we also integrate both the remote sensing derived biophysical variable with some like fundamental ecophysiological mechanism into like a computer model so that we can understand about the climate change impact on the plant ecophysiological processes. That's general ideas about my lab research. So the evolution scientific question my lab try to address is kind of to try to solve the climate change threats. Like multiple colleagues already point out, global warming is already the fact, right? And with the global warming, not only for our natural ecosystem and also for the agricultural ecosystem has been greatly impacted. And I think especially when it touches to the food security issues, it will fundamentally will affect the life and the living quality of billions of people in the world. So that's a huge issue. That's also a key reason today we gather together to think about whether we can solve this kind of problem. And my science to solve this kind of climate change threat are primarily based on the plant ecophysiology theory. And by definition, plant ecophysiology aim to study how plants sense and respond to environmental change, and which ultimately affect their function. And a simple example about the plant ecophysiology probably is the study of the plant photosynthesis. And it's important because through photosynthesis, plant can provide food and feed the entire planet. But for our climate change science, plant also provide really important role to regulate both the local and the regional climate. So that means if we want to really resolve the climate change threat, the first important step is to understand how climate change will affect plant ecophysiology processes. That's also the key hypothesis of my lab research. And the next, I try to use 2 examples to show how the study of the plant ecophysiology can potentially make a global impact. And one is the global food issues like we all discussed here. And like the figure shown here, in 2020, according to World Report, and we know already have more than 30% of people suffer with the food insecurity issues. And over the time, this ratio has been changing. Now, if we project to the future, and we know with rapid population increase, that means we are going to demand more food supply, right, to meet the population demand. But at the same time, with the climate change, it will make more regions probably are not sustainable, right, for agricultural production. That will means this kind of like the percentage actually will increase. And to solve this problem, I really like the paper raised by Jonathan Follis, date back to 2011 in Nature. So he came up a simple and elegant solution for this. Is one thing is definitely we can turn to more land, but this is not a sustainable approach to solve the food security problem. And the other potential is we can increase per area crop yield. And this will definitely call for advanced science and technology. And like Professor Lang already pointed out, modern genome engineering technique can provide one way to solve this kind of problem. But on the other hand, imagine if you have a plant already grows in the real world, and it probably will still subject to various environmental stress, right? And how can we solve this? So modern technology like the sensor technology potentially can bring in to help to solve this kind of problem. And we call this as potential like the precision agriculture technique. So based on this kind of general background, and when I joined CUHK-U and last year, I feel really fortunate to have a conversation with Professor Lam and also Professor Michael Ng, another professor from University of Hong Kong, and he's a big data computer science. He's kind of like a mathematician, but with the experts in both mathematics and also big data analysis, including AI. So we here just label the expertise from each of our key members, and we found if we can bring all this kind of expertise together, actually probably we can do something different. So one thing we can start is try to think about whether we can use the sensor technology, plant ecophysiology theory, plus the machine learning technique to help to solve some fundamental issue attached to the precision agriculture. So we come up with a small seed project called the smart sensing of plant water use. So why we want to start from here? Because we know in a leaf, right, there are thousands of the stomata, right? And during plant photosynthesis, actually through the stomata, plant will trade water transpiration for carbon gain, right? But one thing we know, water are not unlimited resources for plant growth. So that means if we can real-time smart sensing of the plant water use, it can potentially provide a really key component for the modern precision agriculture. So that's the general ideas. We try to team up and also try to solve this problem together. And to do this, now we recently just started with this project, and we divided into multiple different phases. And in the phase one, we try to leverage the lab experiment and try to grow some model plant in the lab conditions and also mitigate in different environmental conditions, and plus using our sensor technology so that we can see whether we can develop this kind of like a smart sensing approach about the plant water use. And in the next phase, we also try to think about whether we can extend it to the real field world. So that, for example, we can mount our sensors also in the drone, and then it can see it in a much bigger landscape. And eventually, we now already have a lot of select remote sensing technique. So with this kind of like a real observation from our ground surveys, and we can also use select remote sensing to think about whether we can scale up to even a large scale. So that's still in the planning. Like I said, I'm still early to the agriculture field. So I really welcome any comment and also feedback on this. And the other part actually are more directly related with my research work. We try to solve the global ecology issue. And we know one thing is the anthropogenic carbon emission is one big driver of the current global warming. And with the increase of the atmospheric CO2, on the one hand, it can potentially be benefit for the plants, right? That's known as the CO2 polarization effect, because CO2 is a really important ingredient for plant photosynthesis. That means with the enhanced carbon photosensitive carbon uptake, plant potentially can remove some CO2 from the atmosphere. So in that sense, that means plant actually can play a really important role in battling the future climate change. But on the other hand, we know with the global warming, with the higher CO2, that means normally it's a global warming. That means plant actually will suffer with the enhanced like the heat and also water stress, and it will lead to enhanced chance of the fire, right, wildfire. And with the fire, that means original carbon stored in the plant will be released back to the atmosphere. And then in this pathway, plant actually play a really important role in accelerate the future climate change. And now we already have a lot of data and evidence shows over the past 30 years, plant pathway 1 dominated by pathway 2, and we, as our human beings, we still can enjoy the benefit from the plant and serving as a really important way to battle climate change. But we are not quite sure what will happen in the future, because already you have several evidence already shows over the last 30 years, now we are shift from the pathway dominant planet to the pathway dominant, pathway 2 dominant planet in the futures. So where is the tipping point, and where is the timing for us to take action so that we can help use the science and technology to help to preserve our planet? That's still a big unknown. So that's the key scientific question now my lab try to address. And how can we do it? So now my lab try to leverage a lot of, we call a big Earth observation. We have a sensor now in the ground, in the drone, and in the airplane, and even in the satellite. So we try to fuse all these kind of sensors together, and then it will definitely generate like a really big data. So we also collaborate with the computer science, so we can generate a high performance computing, and also like a deep learning technique, so we can extract really useful information out to help us to understand what's going on in our planet. And finally, we also incorporate all this kind of information into our models so that we can understand how the plant will be sensitive to the climate change and potentially take solution we can do to help to preserve our planet. So that's the general ideas about my lab research now is working on. And I think some technique, especially like the big data and also the deep learning we develop here potentially can be also useful for the agriculture field. And here is my lab web page and also email contact. If you are interested, feel free to contact me. And also thank everyone and look forward to more conversation and collaboration in the future. Yeah, thank you.
Thank you, Professor Wu. I think your research is very interesting and applicable to agriculture because we are dealing with a lot of soil problem, vegetation, deforesting, different kind of crops. So we can do remote sensing using big data and using high spectrum analysis. Maybe it will speed up the whole process rather than sending students to do the survey inch by inch. So I think we will talk more in the discussion sessions.
Yeah.
Now, come to our last talk. Right. So last but not least, Professor Dr. Khalid Safala is Han is the Professor of Soil Science of Institutes of Soil and Environmental Sciences at PMSA Agricultural University. His research interests include the study of soil microbes, biomass, and biochemical parameters as an indicator of changes in soil system. Let's invite Professor Han to share with us on agriculture, environmentalization, and its impact on soil productivity, please.
Good afternoon, everybody. And thank you very much, Professor Lam, for my introduction. First of all, I appreciate the organizers of today's webinar for bringing the scientists from Hong Kong, China, and Pakistan together. Since I'm representing the Institute of Soil and Environmental Sciences from PMAS, Arid Agriculture University, Rawalpindi, you know, our university lies in the northern part of the Punjab province in Rawalpindi city, which is very, very close to almost, you know, very much connected with the capital of the Pakistan, Islamabad. The mandate of our university is to deal with the agricultural issues of rain-fed areas of Pakistan. And the rain-fed area in our country constitute almost 25% of the total cultivated area of the country, which is 20.61 million hectares. And again, 1/4 of the total geographical area of Pakistan, that is 79.61 million hectares. Earlier speaking, Dr. Ghulam Murtaza and Dr. Zahir Ahmed Zahir from University of Agriculture, Faisalabad, since both of them are also the professors of soils, so they have very nicely highlighted the issues of soils of Pakistan and their management practices. My focus will be on the issues of the soils which are under dry land agriculture or the rain-fed agriculture. So if we talk about agricultural sustainability, soil sustainability, and environmental sustainability, then all these aspects are very much dependent on each other. And long-term food security of any nation lies on the effective handling of the agro-ecosystem and use of environment-friendly technologies. So keeping this in view, I would like to share with you a threefold relationship between agriculture and the environment. The first and foremost is that agriculture is a contributor to environmental deterioration in several ways. For example, greenhouse gas emissions from agricultural lands, and then groundwater pollution due to intensive use of fertilizers, and also the loss of organic matter, and contributing to other environmental issues. But on the same time, the impact of environmental deterioration on agriculture is also great. For example, if the concentration of heavy metals and other toxic elements is increasing in terms of industrial waste, and those are applied to agricultural lands, so our lands get polluted or contaminated, and that will also deteriorate the food chain of our and will have a negative impact on the health of the people which are feeding upon that food produced from the polluted lands. Similarly, agriculture is also a potential solution of environmental deterioration. For example, the waste materials are produced, and the best solution for the available for the disposal of the waste is the land application, but in a scientific way, not in an unscientific or injudicious way. So as far as environmental stress and soil productivity issues are concerned, the three major environmental stresses, for example, climatic variability, industrial activities, and over-exploitation. I will just want to mention about the effects of these environmental stresses. For example, climatic variability. It is usually expressed in terms of the erratic rainfall and the rise in temperature that will result eventually into water stress and loss of nutrients and depletion of soil organic matter. Similarly, industrial activities generate waste material which contains toxic organic and inorganic pollutants, and when this waste is added or applied, then it increases the toxicity and reduces productivity of the agricultural land. And likewise, over-exploitation of the resources, for example, overcropping, intensive cropping, intensive tillage, and the overgrazing, this results eventually into nutrient exhaustion and depletion of the nutrient resources of the soil, and eventually this all leads to soil degradation. As far as soils of Pakistan are concerned, the Pakistan is predominantly an arid to semi-arid country with 80% of the area receiving rainfall less than 300 millimeters. So very much, dry area, and the annual precipitation is less as compared to evapotranspiration losses from the soil and the land surface. So 1/4 of country's land is suitable for agriculture, but is threatened by fertility loss, wind and water erosion, salinization, sodification, waterlogging, and loss of organic matter. The details I will not discuss because earlier speakers, they have focused on this. I would just like to focus on environmental issues and their solutions. And if we talk about, for example, the resource recovery and utilization as a resource, over exploitation of the resources, for example, the sustainable use of resources can be a solution to this, and proper management can also be the solution of the over exploitation, because that will eventually be resulting into degradation and deterioration of the soil resources, as earlier said by Dr. Ghulam Murtaza, that 1 centimeter layer of soil, it takes almost 500 years to form. So therefore, the soil is a very much finite resource and very precious resource. And if that over exploitation of this resource is carried out, so that will result into the loss of biodiversity and other essential ecosystem functions of the soil, resulting into the poor productivity. Similarly, soil erosion. Soil erosion is also a common issue, especially in the rain-fed areas where water erosion prevails. And this issue can be overcome by terracing, by contouring, by the use and the wind erosion, by the use of the shelter belts, then continuous vegetative cover. If we maintain, then we will be protecting our soil resources against the erosion. Similarly, the increase in soil organic matter, which is very, very important for a very productive land, or for the soil to be optimum productive, at least 2% organic matter is required. But Pakistani soils, they have less than 1% organic matter, and particularly majority of the soils in the rain-fed areas, they are very, very low in organic matter content, which is less than 0.5% in most of the cases. So land degradation, so we have to adopt the management practices which result into the improvement of the soil productivity rather than leading to deterioration of the soil. So we can monitor soil health at regular intervals of time, and then we can relate the changes in the soil health conditions with the management practices which keep on going or which are being practiced. And then in view of the positive effect of the management practices, we can adopt those practices on long-term basis so that the land degradation can be avoided and the sustainable use of the land resources can be managed. Then nutrient loss, for that we can go for organic fertilization, we can go for judicious use of chemical fertilizers, and this way we can compensate for the nutrient loss from the soils. Similarly, integrated plant nutrient management technique can be adopted and can be used for sustaining long-term productivity of the soil. So next is the climatic extremes. So in this regard, the early warning systems can be developed, and which can inform us, we can tell us about the changes that are taking place in the climatic conditions and weather conditions, so that the appropriate measures can be taken for maintaining the productivity of the land resources. Similarly, utilization of climate insensitive varieties. You know, as earlier has been discussed, that the crop growth period has been shortened because of the climate change effects, and Pakistan is one of the top few countries which are vulnerable to climate change effects. So therefore, those varieties can be used, crop varieties which are insensitive to changes can be developed. And carbon sequestration, as I said, there is loss of organic matter from the soil. So we should also adopt such management practices that will eventually lead to carbon sequestration and improvement of soil organic matter content and improvement of the overall productivity of our land resources. So what type of work is going on at the Institute of Soil and Environmental Sciences in our university? I would like to share very briefly about that. The first and foremost issue in the rain-fed agriculture, especially the Potohar Plateau, where our university lies, is the soil erosion and runoff losses from the torrential heavy rains, especially in the monsoon season. So that is resulting into the loss of precious land resource, soil resources. And in this regard, we are carrying out the joint research efforts with the West Agriculture and Forestry University of China for the measurement of soil erosion and runoff in this area. So different slopes have been prepared at the research farm of the university, and then automatic gadgets for the measurement of soil erosion and runoff losses have been installed. And also we are using different land use practices like the bushes, the forests, and the crops, and then different types of crops, and then different types of crop growing systems like contour cultivation, and along the slope cultivation and across the slope cultivation. So this type of long-term experiment is set up over the farm of our university, and this type of experiments are being carried out. Similarly, we are also working a lot in the form of a long-term research trial on tillage effects on soil health. So different types of tillage systems are being adopted. For example, the conventional tillage system, the zero tillage system, the minimum tillage system, and the modified reduced tillage system. So effect of different tillage systems on long-term soil productivity is being assessed, especially using the century model. And in this regard, that it has been revealed that under the conditions of rain-fed agriculture, the reduced tillage is most effective and leading to buildup of soil organic carbon and improvement of soil health and soil fertility status. Similarly, the other issue, as I said, is the less organic matter contents in the soil. So for that, the use of recalcitrant organic materials for improving soil organic matter contents is this type of work is going on. For example, the use of biochar materials and different types of biochars prepared from different feedstocks, and what is their effect on carbon sequestration, what is their effect on soil physical properties, what is their effect on soil chemical properties, for example, cation exchange capacity, and what is their effect on soil microbial and biochemical properties, which are really essential for maintaining the soil health. So the use of recalcitrant organic matter, our institute is leading in this type of research. Similarly, we have also prepared slow release nitrogen fertilizers by coating the urea with different materials. As the nitrogen use efficiency is very low almost all over the world, and especially in Pakistan, it is around 30%, and 70% of the nitrogen fertilizer are lost from agricultural lands due to high temperature, due to alkalinity of the soil, and also due to application of urea as a single dose. So then if we are using the slow release and fertilizers, then definitely this will lead to the better nitrogen use efficiency in soil plant system. Similarly, we have also conducted research in case of nitrogen inhibition processes. So different chemicals, organic and inorganic in nature, have been tried for checking down the rate of nitrification, because the nitrates are lost from the agricultural field in the form of leaching and in the form of denitrification. So to check those losses, the use of nitrification inhibitors is effective, and this type of work we are also carrying out. Similarly, the potential of plant growth promoting rhizobacteria and biofertilizers under the rain-fed agriculture, long-term trials have been established, and different microbial strains have been isolated from the rhizosphere of different crops grown under the rain-fed conditions, and then their effect on water conservation under the rain-fed conditions, and on improving the nutrient availability in soil under the rain-fed condition, and improving crop yields has been evaluated. Similarly, soil microbial biomass as a source and sink of plant nutrition. This kind of research trials have also been carried out, and work is still going on, that how can we improve the microbial biomass, which is an important indicator of soil health, an important indicator of the productivity of agricultural soils can be improved and can be used for as a source of nutrient for the plants, and their role, especially in the degradation and decomposition of organic materials added to soil, can be utilized. And similarly, monitoring of greenhouse gas emissions from agricultural lands. So since the temperature is high and the rainfall is low, the overall precipitation is low, and agricultural fields, they are contributing particularly to methane emissions, carbon dioxide emissions due to enhanced microbial activity under the conditions prevailing in our system, and likewise the nitrous oxide emissions from agricultural fields. So we are monitoring the greenhouse gas emissions, and then we are also carrying out the practices that how can we minimize the emissions of greenhouse gases from our soil. So organic waste management is another important aspect on which we are carrying out the research. There is significant work going on composting and vermicomposting, and also conversion of organic waste into biochar and use of these recalcitrant organic materials for improving soil conditions is being carried out. Similarly, the soil pollution from pesticides and industrial effluents, especially in the area around heavy mechanical complex, the industrial effluents are released, and those effluents eventually enter into the agricultural lands, and then they are contaminating these agricultural lands, and they can also be translocated into the crop plants and therefore can enter the food chain. So keeping in view all this, so work is also going on that how can we decontaminate, especially we can reduce the availability of toxic elements in soil by the use of biochar and other soil amendments. And being also the Director of Research, Innovation and Commercialization of our university, I will also like to share that our university is also working on digital soil mapping and variable rate nutrient application technology for precision agriculture. Similarly, we have also working on the industrial biotechnology and other aspects, and also the livestock genomics and related issues. So I would very much welcome the colleagues and scientists, fellow scientists from mainland China and from Hong Kong to work with our scientists from our university for improving the overall agricultural productivity in the Southeast Asia and in the South Asian region, especially in Pakistan and China. Thank you very much once again for giving me time to share my research and the activities being carried out in our university with all the scientists, fellow scientists. Thank you very much.
Thank you. Thank you very much. I think it has been a long day and all the speakers has give excellent talks, and I believe this exchange is very fruitful for both sides. So I'm looking forward for future collaborations, and hopefully next time we should meet face to face, either in Pakistan or in Hong Kong. We'll be very happy to visit the field in Pakistan. I know that there's a lot of beautiful areas because I used to have a postdoc who's from Pakistan. He said that some of the villages are very beautiful and with a lot of crops that we can visit. So maybe after the pandemic, we all visit Pakistan someday and start our collaboration. Okay.
Definitely.
All right. So with that, I thank everybody, and I think good afternoon, everybody. So we'll see each other soon. Okay.
Thank you very much. Thank you, all of you, sir.
Bye bye.
Thank you from my side. Thank you.
Thank you, sir.
Bye bye. Bye bye.
Bye bye.